Hi everyone, my name is Gilles Gouspillou and I'm going to present our recent work today on Parkin-mediated mitophagy in skeletal muscle health and aging. But first of all, I want to thank the organizing committee for allowing me to present our work. So I'll start with presentation by providing a very brief introduction of what muscle aging is and looks like. On this slide, just right here, what you can see is two MRI images of the thighs of a young individual on the left and an old individual on the right. In gray, you can see the muscle. And what is very easy to depict here is that in the old leg, we can see that there is less muscle. We can also see that there is more white, which is a sign of fat infiltration. So this is a clear sign here that with aging, we have a loss of muscle mass. What is very logical is that with the decline in muscle mass, we also get a decline in muscle strength or muscle force. What is really unfortunate with aging is that whatever we have left of muscle doesn't work as well. So we have an impaired quality of contraction. So these three phenomena combined, decline in muscle mass, decline in muscle force, and impaired quality of contraction, defines what we call sarcopenia. So sarcopenia is a very, very complex and multifactorial process that can be influenced by many things. There are many mechanisms that have been proposed to contribute to sarcopenia. Just to name a few: hormonal changes that occur with aging, neurological alterations that develop, systemic inflammation, pathologies can impact the development of sarcopenia, changes in physical activity and nutritional intake can also either worsen or to some extent ameliorate sarcopenia. So this is a very, very complex phenomenon. But what I'm going to focus on today is that there is pretty solid evidence now in the literature and consensus that mitochondrial dysfunction do play a role in the sarcopenic process. So to explain to you what these mitochondrial dysfunctions are, I'm going to use this slide and I'm going to start to briefly explain what you can see on this slide. So here you can see the mitochondrion with the outer membrane and the inner membrane. You can see the complex 1, 2, 3, and 4. This is the electron transfer system. You have the ATP synthase right there that is there to produce ATP. So all of this together is the oxidative phosphorylation. Through its normal functioning, this system here, the electron transfer system, will produce superoxide anion, which is a free radical. If this is produced in excess, it can lead to oxidative stress. Another aspect that I want to touch upon here is this pore here, the mitochondrial permeability transition pore, which is a pore that can regulate mitochondrial-mediated apoptosis. This pore can be open if mitochondria are overloaded with calcium, or if there are too many superoxide anion produced or too much oxidative stress in the mitochondrial matrix. So two potential trigger here, too much free radical or too much calcium inside the matrix can open the MPTP. So with aging, what has been proposed is that, and it's a theory that is still there, but has been quite challenged in the past decades, which postulated that with aging, we get an increase in ROS production. So this is why you get a question mark here. Why I did a question mark is that there is still debate on whether or not mitochondrial ROS overproduction is involved, but I mention it because it's in the field here. So this could be a contributor. So too many ROS production causing damages to many components in the mitochondria, including the oxidative phosphorylation and the MPTP, which could, of course, cause a lot of problems for mitochondrial function. A consensus has emerged on the impact of aging on mitochondrial energetics. Like the vast majority of studies show that aging is associated with a decline in ATP production, mitochondrial ATP production. Another aspect that also makes consensus is that the function of this pore, the mitochondrial permeability transition pore here, is altered in aged skeletal muscle. And one of the consequences of this alteration is that there is an increase in mitochondrial mediated apoptosis, which is something reported in across all studies that have assessed mitochondrial mediated apoptosis in the field of muscle aging. So although ROS production is in question these days, there are clear evidence that mitochondrial dysfunction do contribute to the loss of muscle mass and functionality with aging. One question that remains unanswered as of today is, but what is the mechanism? Why do old muscle accumulate mitochondrial dysfunction? And this question is very legitimate because in our cells, we do dispose of systems to ensure mitochondrial quality control. And one of them is Parkin-mediated mitophagy. So mitophagy is a process where dysfunctional mitochondria are useless mitochondrion will be degraded through the process of autophagy. So this is depicted here, where you have like a normal healthy mitochondrion, which becomes damaged or dysfunctional. You can see that this process that I present here involves pink one, Parkin. So this is the Parkin-mediated mitophagy that I'm presenting here. And what is going to happen is that the dysfunctional mitochondria are going to be degraded through this process. So today, my talk is only going to be focused on Parkin-mediated mitophagy. I just want you to know that there are many pathways that control mitophagy. For the sake of time, I can only focus on Parkin-mediated mitophagy today. So Parkin is an E3 ligase encoded by the Park2 gene. It regulates mitophagy by ubiquitinating outer mitochondrial proteins. And once they are ubiquitinated, these proteins, and several of them are ubiquitinated on the outer membrane, then the mitochondrion in question will be targeted to autophagy. To do so, Parkin has to interact with two proteins, which is pink1 and VDAC. So VDAC, which just appears here, which has been shown, which has been reported to serve as a docking platform for Parkin. So, as my title mentions here, is that a few years back, we stated this hypothesis that maybe one of the mechanisms that could contribute to the accumulation of mitochondrial dysfunction with aging could be a defect in mitophagy, and specifically in Parkin-mediated mitophagy. To start testing this hypothesis, what we did at the time is we were lucky enough to have access to biopsies, so human biopsies, from young and old participants. And in these old participants, we knew already, and we published it in this paper that is highlighted here, that there was muscle atrophy and there was mitochondrial dysfunction. And what we wanted to know in this population is what is happening to the Parkin to VDAC ratio. So Parkin needs to bind to VDAC to act. So we probe for this ratio here as an index of mitophagy. And what we found here is a like very preliminary, but the first evidence that maybe there is something wrong with Parkin-mediated mitophagy with human skeletal muscle aging, because this ratio, as you can see here, is decreased, significantly decreased in our old individuals. So another piece of data that's really pushed us to study the role that Parkin plays in skeletal muscle is that these same individuals that I described before, we did a lot of things with their biopsies, and we also tested them quite thoroughly in vivo. Apologies. So we assessed the fiber cross-sectional area, their leg power, their performance at 3 different functional tests: the time up and go, stair test, and sit to stand. And what you can see here is that the Parkin to VDAC ratio correlated with all of these measures. And this to us prompted us to keep working on Parkin and the Parkin-mediated mitophagy pathway, because clearly this association suggested to us that this protein might be important for skeletal muscles. And we specifically asked 3 questions. First, what are the roles of Parkin in skeletal muscle, normal skeletal muscle? Can Parkin overexpression attenuate sarcopenia? And can Parkin overexpression attenuate sepsis-induced muscle dysfunction? Sepsis being a condition that often can affect the elderly, so which is very prevalent in the elderly, or quite prevalent. So to answer our first question, or to define the role of Parkin in normal skeletal muscle, we used Park2 knockout mice. So that's mice that are deficient in Parkin, as you can see here, like Parkin is absent in skeletal muscle and in other tissues. The first thing we did is that we assessed the in situ contractility, so the impact of Parkin ablation on the in situ contractility. And what we found is that there was a mild but significant alteration in muscle contractility. Maximal strength was reduced in Parkin knockout animals. To kind of probe for potential mechanisms explaining why muscle strength was decreased, we assessed muscle fiber size. To our surprise, Parkin knockout did not result in muscle atrophy, as you can see here. So there was no muscle atrophy that we could detect in these young animals. We next assess mitochondrial respiration. I don't have the time to go through the protocol, but basically here, this is maximal respiration when we only provide complex 1 substrate. And this is a test that assesses the maximal respiration of the mitochondrion. And what you can see here, very briefly, is that Parkin knockout results in severe mitochondrial dysfunction, decrease in respiration. It's a pretty severe reduction that we see here. We assessed what is called the control ratio, which is an index of mitochondrial coupling efficiency, how good mitochondria are to produce ATP. And you can see that there is a dramatic decline here in this ACR ratio, indicating that mitochondria are really dysfunctional. When we probe for the enzymatic activity of citrate synthase, complex 2, SDH, complex 1, and COX, complex 4 here, we found that some enzymes, but not all, were their activity was reduced in Parkin knockout animals. So clearly here, there was signs of mitochondrial dysfunction in Parkin knockout animals, saying that Parkin is very important for skeletal muscles. To our surprise, we used a complex protocol to assess ROS production that we measured through H2O2 emission. We did not see any impact of Parkin ablation on ROS production. However, we found that this marker here, 4-HNE, which is a marker of oxidative stress, was increased upon Parkin knockdown. So to further our analysis of the impact of Parkin ablation skeletal muscle, we tested the impact on the MPTP function, which again regulates a process known as apoptosis. What we did is that we challenged mitochondria with calcium to see how fast this pore was opening. And what we found is that there was a significant decrease in the time to pore opening, indicating that the MPTP is dysfunctional in Parkin knockout animal. So to sum up this first study, we showed that Parkin ablation causes mild impairment in contractility, severely impairs mitochondrial respiration, and sensitizes the MPTP. So these results taken all together clearly indicate that Parkin is essential for normal skeletal muscle function. So we then moved on to test whether Parkin could be overexpressed to slow down the muscle aging process. And to do this, what we've done is that we use adeno-associated viruses injection. So we injected one leg of our animals, the muscles of one leg, with an AAV containing the sequence coding for Parkin, and the other leg, the contralateral leg, with a sequence coding for GFP, so a control protein. We studied two groups, young animals. We started at 3 months and we overexpressed Parkin for 4 months. We studied them at 7 months. For the old group, we started at an age where sarcopenia is not there yet. And so 18 months of age, and we overexpressed Parkin for 4 months. And we studied these animals 22 months, where we know sarcopenia is established at the time. So this is just to show you quick Western blots to show you that we were successful in overexpressing Parkin in both young and old animals. And this is where it gets really interesting. So when you take this group here, the green, that's the young muscle. So this is the tibialis anterior weight. If you compare it to the control, so GFP expressing muscle of the old animals, we can see that there is a muscle atrophy. So our animals were sarcopenic. And what you can see here is the impact from green to blue of Parkin overexpression in young and old animals here. What was very, very interesting is that in both young and old animals, Parkin overexpression increased muscle mass. This was associated with, in all animals, we probed for it, an increase in muscle strength. So Parkin increased muscle mass and muscle strength. We then probed here, it's muscle cross-section stain for 16A dehydrogenase activity in all of our group. What we found is that Parkin overexpression in both young and old animals increased the activity of 16A dehydrogenase. And we used transmission electron microscopy to assess mitochondrial density in all animals and the impact of Parkin overexpression. And we found that Parkin overexpression increased mitochondrial density in all skeletal muscle. To assess the impact of Parkin overexpression on apoptosis, which we know is increased with aging, and we can see it here, we use the tunnel stain, which is sensitive to apoptosis. And we can see here that with normal aging, there is an increase in the number of tunnel positive myonuclei. And if you overexpress Parkin in the old, you decrease this number, so you decrease apoptosis. So just to sum up this second study here, overexpressing Parkin attenuated sarcopenia, increased muscle mass in young animals, increased markers of mitochondrial content, and attenuated apoptosis in all skeletal muscle. So I want to leave you here with this message that increasing Parkin expression in all muscle could represent a promising therapeutic target to counteract sarcopenia. So just to briefly, it's very quick to touch upon sepsis-induced muscle dysfunction. This is just to show you that septic patients develop skeletal muscle weakness. You can see it on this graph and you can see it here. Septic animals, so where sepsis is induced, display in at different time points here muscle atrophy. So you do get muscle weakness, muscle atrophy, And very quickly here, that's the mitochondria in normal muscle. That's mitochondria in LPS treated animals. So LPS mimicking sepsis in this condition. And you can see that sepsis induces mitochondrial dysfunction. Abnormal mitochondria are present in sepsis. So we use the same approach. We overexpress Parkin using our AAVs in septic animals. To achieve sepsis, what we've done is we use the Cecal, ligation, and puncture model, CLP model, which basically you take the cecum, you ligate it, puncture, and put it back in the abdomen. Sepsis will develop very rapidly with these methods. And so this is just to show you that Parkin overexpression was successful in our model. Parkin was overexpressed in our muscles. Now, what I want to show you where it gets interesting is that similarly to what I've shown before, overexpressing Parkin results in hypertrophy in controlled, so sham-operated animals. But what is very interesting is that you can see the very nice effect here on muscle fiber size of Parkin overexpression. So Parkin had a positive impact on fiber size in septic animals. So these are the septic one, the control and Parkin overexpressor here. Very interestingly, and I'll go very briefly here to not go over time, what you can see here is that for complex 1 and complex 4, the two here, Parkin overexpression attenuated the impact of sepsis. So Parkin overexpression preserved complex 1 and complex 4 content. So to sum up our third and last study, in septic skeletal muscle, overexpressing Parkin resulted in larger myofibers and preserved mitochondrial protein content. So similarly to my previous conclusion, potentially overexpressing Parkin could represent a therapeutic target to attenuate sepsis-induced muscle and mitochondrial dysfunction. And with that being said, I just want to acknowledge all the people that did the work, all the students that were involved and collaborators that made this work possible, and all the funding sources. I thank you very much for your attention.